Okay, Roger Mudfossil University, excited as can be. We're going to be looking at a presentation by California University talking about bacteria in the gut, and they're now doing the research that I was hoping somebody would do, and they are going to do it. So bacteria is number one. It's the most number one thing in your whole body. Exists on and in you everywhere. It lives in the membranes. It creates mucus and enzymes. The enzymes create biomolecules, which you need to produce all the things in your body to live. The mucus protects you, and it protects themselves at the same time. It's a cooperative sort of effort. Bacteria also is related to your emotions. All kinds of problems from bacteria. And they have fully understand, well, they don't fully understand it, but they understand fully that these things occur. Now, bottom line is bacteria is your workforce and it is your protector. It goes and it breaks up molecules, it creates all the products you need, it creates a transition metal, ligand attachments, and all that sort of stuff. Now, it is your protector. It protects you against other stuff to come in to attack you. It creates barriers with mucus. It lives in the membranes and it feeds them with what it ends up pooping. It creates enzymes that create molecules. Enzymes are nothing more than little chemistry sets. They're not alive. They go out and they crack molecules. Every bacteria basically creates one, and it has a job to do. And if the bacteria is not there, you don't have the enzyme is not there, you don't make the molecules. Or if the molecules aren't there to start with, the raw materials, then you're not, I don't care how much bacteria you got, you're not going to make them. So it's a cooperative effort. Now, good fights bad bacteria. So just because you got bacteria in you, that doesn't mean it's bad. You have to have bacteria literally everywhere in your body, on your surface, everywhere, all over your eyeballs, everywhere. Now, sometimes you can get bad, yes, absolutely. But normally it will only be bad if it's attacking because the good is not there to protect you. All right, so let's go a little further. Okay, remember I said there's a lot of membrane layers? Well, this is a membrane, and these are the kind of layers that are in there. And in between every one of these layers is bacteria living in there. And it creates a slime layer. And inside there creates the enzymes that the body needs to break down the molecules so that it can live. Now, this is the University of California. Very, very good. I'm really, really excited here. Here she goes. Human microbiome. This is what we're talking about, gut bacteria.
I'm thrilled to be with you tonight to share with you a relatively new and I think incredibly exciting field of research that's really changing our view of human biology, the field of human microbiome research. And that really describes studying the intense diverse communities of bacteria, fungi, and viruses that live in and on the human body and understanding how they shape our health. So what will we cover tonight? Well, first things first, how do you study entire communities of microbes? What tools do we have, and how do we understand not just who's there, but what they're doing and how they're interacting with the human host? Then we'll go through a quick whirlwind of the human microbiome 101.
You know, I'm not going to go through, she's going to go into some very, very good things. And I like her. I want to try to contact her. But what we need to know is what microbes exist, where they exist in the body, how much the quantities of them are in those specific areas. Then in like autopsy results and things from healthy people and unhealthy people. And then we can determine what is it, is there a difference? Obviously there's going to be some form of difference. In some form, you know, is your kidney have some missing chemistry that needs to be there to break down the salts or to work with the salts, or I mean, there's just so much that can be gleaned just from what's there. You look at what's there, and then you determine what you got to do why the other one isn't there. I mean, this is exciting, very, very, very exciting. So anyway, I'm going to just leave it. Well, I'm sure I won't leave it at this, but at this point, we've got a way to move forward. We need to find out what exists, where it is, and then we'll know what a healthy human being is. Right now, we just don't even know. I'm not kidding you. They just been paying attention to this. They sent you home and they take some antibiotics, go home. Then they decided, well, maybe that's not a real good idea. Maybe we should do something. But by that time, a lot of people were damaged, and I mean damaged. All right, now she's already made the statements that bacteria is a major thing in your entire body. So here we go.
Tools for assessment of the microbiome have rapidly evolved and expanded in capacity over the last several years.
All right, before she goes on, I want to make a statement. I have been saying right along that we have to treat this exactly identical to the Human Genome Project, where we went in and studied all the bits and pieces in that sequencing of the human genome and determined what bits and pieces were there. Well, now we understand the bacteria, and now they have the tools to determine what lives in every niche in your system. And you have a zillion, your intestines and your colon and your stomach and your throat and your lungs and your mouth and your esophagus, they're all different. All different bacteria, doing different jobs, protecting you from different invasive things. Now, let's let her talk.
Years. And we now can, instead of just looking at a biomarker gene, actually take all of the DNA we extracted and sequence all of that DNA. And then put those pieces back together, basically reassembling the genomes of all of the microbes in that sample.
All right, so we have a human genome now in us. We know what particles are supposed to be in what arrangements. Well, now they're able to go in and say, look at all this bacteria. There's 1,000 different bacteria there. Well, we know all the different DNA of all those different bacteria, or at least they're going to learn them. And at that point, they can say, we have so much of this DNA, so much of that DNA, and quantities. And then they can determine what that biome literally consists of.
And this is no small feat, as you can imagine.
No small feat
I
at
joke,
all.
although it's not really a joke, and I say it's like me handing you War and Peace by Tolstoy in pieces and asking you to put it all back together in a legible form. That's the computational capacity that we need is immense to do this job, but something that we have developed very rapidly over the last several years. So while biomarker gene sequencing tells us who's there, shotgun metagenomics tells us the genes that these organisms encode and what they have the capacity to do. But we've pushed this field even further. We can also extract RNA from a sample. It's another type of nucleic acid, and it's the transcription of those genes. So it's what is that community of microbes actually transcribing off their genomes? How are they responding to the current conditions? And we can sequence those pools of extracted RNA by sequencing also, and we call this metatranscriptomics. It gives us a snapshot into the genes that are being expressed at the time of sample collection by organisms in the microbiome.
All right, this is unbelievable because, right now they think that the genes, they just transcribe a certain set of these different molecules, CTAGs, and they're different. CT and A and G all stand for different molecules, and they think they just transcribe. That's it. That's that code every time. If it's not that code, you're sick. Well, apparently, it changes. It depends on how you are, and you may be sick. But it's not necessarily that the transcription of it's not necessarily the code is just broken forever. It's just that at that time you're sick and the code is being affected. It sounds to me.
But what's even more exciting is that we can look even deeper. These are all next generation sequencing based tools to look at microbiomes. We can also use mass spectrometry, the capacity to identify small molecules. And we can use this to look at protein pools produced by the microbiome, to understand the proteins that they produce. And that includes, remember, all of the enzymes and catalytic functions.
Enzymes and catalytic functions. Enzymes are the things that break things. Catalysts are the things that facilitate fast reactions.
Of the microbiome. But what's nearest and dearest to my heart is metabolomics, looking at the small molecules. Remember I said that that's how microbes communicate with each other. In fact, that's how cells communicate with each other, irrespective of their microbial or host.
Now, this is something also that I haven't looked into deeply myself, because how do they communicate? How is that signal sent? How does it interpret the signal and make sense of it? Well, guess what? I found out today they cut a mouse brain in half, 2 pieces, and they could still communicate to each other. This could send the signals of this brain. Somehow it got the signal across nothing, through the air. So every day it's something new. And I think she understands this. I like this girl, lady.
And this, for me, is the lexicon that governs microbial host-human host interactions. And this is where we think the next frontier, and we're already realizing the next frontier in this field really lies. So the application of these tools, and in particular DNA-based tools, has massively expanded our view of bacterial life on this planet.
By the way, I'm going to probably leave it at this, but you should watch this. It's an hour and a half long almost, so it's going to take a while. But she seems to be a fountain of information. Now, I haven't watched it totally myself either. But I did send her an email, and I copied Andy, who is Andy Marino, who owns a university that installs bacteriological DNA labs into food service facilities, and they train the people, and they go out and they actually install them, and I support them. I imagine it's a service them and all that. So that way there they can get the food supply right out in a matter of hours by using DNA instead of doing all these cultures and things. So we'd love to engage with her, and I did get a reply back that said she's not going to be around till the 3rd of January. Maybe we'll get together. I don't know. I'm hoping. Because I do have some information that from people I've been working with for years, and they have anecdotal stories to tell of here's what I did and here's what happened. And then here's what happened after that. My child got sick again, and then here was the response. All these kind of things are very, very, very important, and they're almost not well done by doctors anymore. There's no country doctor sits around and says, well, how's the kid feeling? Well, how did he play today? And, how does he get along with the dog? I mean, there's so much to a person's life that, to just say, well, he doesn't look too good here, take a couple of these and see what happens. And I know that's not really what happens, and I know they want to help, and I know it's everybody's against everything now. Everybody's against the wall. It's a tough, tough world to live in. Just trying to make things a little easier for some of the weakest, that's all. And I think she is too. I really do. I just have a real good feeling now. A new view of the tree of life. All right? Let's see what happens. It's going to be a new year, 2020. We're all going to have 2020 vision. You got to cover your eyes with mud, and then you get 2020 vision, mud fossil vision. All right? Coming up, 2020.